Hello everyone. So today we are going to look at what are the various factors which affect enzyme action. So let us begin. So these are the factors which affect enzyme action. Okay, and they are first the enzyme concentration, then we have substrate concentration, product concentration, temperature, pH, presence of activators, inhibitors, presence of repressors and derepressors, and covalent modification. So these are the various factors which can affect the action of an enzyme and formation of product. So let us look at the first factor, that is effect of enzyme concentration. Now the rate of the reaction is directly proportional to the enzyme concentration. That means if we look at the graph, and on the x-axis if we have the enzyme concentration, and on the y-axis we have the rate of the reaction, the velocity, then as we increase the enzyme concentration, the rate of the reaction increases. Okay, so it has a direct proportion to the enzyme action. Okay. Now this property is used in lab for estimating various enzymes in the blood. Okay. For example, in a normal person, if we take a blood sample for the enzyme ALT, which is alanine aminotransferase, it's one of the marker for liver dysfunction. Now when we take the blood sample, and the enzyme which is present in it is made to react and form a product. Okay. So this is under normal condition. But when we look at a disease condition, for example, a disease of the liver, so in that condition, the enzyme which is present in the blood is higher. Okay, for example, let us assume it is 10 times higher. Now, when this 10 times higher enzyme is present in the blood, and that is made to react with the undergo a reaction, so the product also which will be formed is 10 times more. Okay, so by estimating the product, it helps us to estimate the enzyme. So this is the principle which is used to estimate enzymes in the blood. Okay, so which is based on the effect of enzyme concentration on the rate of the reaction. Okay, now let us look at the second effect, that is the effect of substrate concentration. Now as the substrate concentration is increased, the velocity of the reaction or the rate of the reaction is increased initially, then it flattens out and doesn't increase with further increase in the substrate concentration. Okay, so again on the x-axis we have the substrate concentration, y-axis we have the velocity. So as the substrate concentration is increased, the rate of the reaction first initially increases, then it flattens out, and with further increase in the substrate concentration, it does not increase the rate of the reaction. Okay, so this can be depicted again with another graph. Okay, we have four conditions A, B, C, and D. So at level, or let's say at condition A, when the low substrate concentration is there, okay, the rate of the reaction is directly proportional, it increases. Okay, now at B, which is half the maximum velocity. Okay, so let us look at the maximum velocity. So C is the maximum velocity. So look at the maximum velocity, all the enzymes over here. Okay, so these C-shaped yellow ones, these are the enzymes, and these red dots, these are the substrate. Now all the enzymes are saturated. Okay, so that means none of the enzymes are free. Okay, so the reaction is at its maximum capacity, maximum velocity. Okay, so this is at position C. Now if you look at position B, it is half of the maximum velocity. Okay, so this is at position B is the enzyme concentration at half the maximum velocity. Now this is used to find out the Km constant or Michaelis-Menten constant. Okay, so the question is, what is Michaelis-Menten constant? It is the substrate concentration at half the maximum velocity. Okay. Now if you look at position D, if you further increase the substrate, the rate of the reaction does not increase further. Why? Because all the enzymes are saturated. So we have this much only enzymes, and all enzymes are already saturated with the substrate. Hence, the rate of the reaction does not increase further on increasing the substrate concentration. Okay. Similarly, we have Lineweaver-Burke plot. Okay, so and the purpose of this plot is to find out the Km value. Okay, so this is the Lineweaver-Burke plot. When is this plot used? When is this graph used? When the substrate concentration is so high that it is practical to achieve that concentration to calculate the Km value. Okay, so then we use this plot. Okay, so where we use reciprocals of lower concentration, and this is used to calculate the Vmax and the Michaelis-Menten constant. Okay, so this is known as Lineweaver-Burke plot. Now let us look at the next factor, which is effect of concentration of the products. Okay, now it is in a reversible reaction. If the product concentration is increased, then the reaction rate is either slowed, it can be stopped, or it may even reverse. Okay, so we have substrate, the enzyme acts on the substrate, we have formulation of product. If the product concentration is increased, depending upon the reaction whether it is reversible, then this whole reaction, it can either slow down, it can stop, or it may even reverse. Okay, so this is the effect of product on the enzyme. Next is the effect of temperature on enzyme action. Now, with increase in temperature, the rate of the reaction increases initially. Okay, increase activation energy and more collision. So if we again on the x-axis we have temperature, on the y-axis we have the velocity of the reaction. So as the temperature is increased, initially the rate of the reaction also increases. Why? Because there is increase in the activation energy and more collision of substrates take place. Then it reaches a maximum point, which is the optimum point. Here this is the maximum velocity that the enzyme has reached, and further increasing the temperature, the rate of the reaction of enzyme decreases. Why? Because the enzymes get denatured with heat. Okay, so if you remember from the previous lecture, enzymes are proteins and proteins become denatured on heating. Okay, so when we increase the temperature further to the optimum temperature, then the proteins become denatured, the enzymes become denatured, they lose their function, and the rate of the reaction decreases. Okay, so this type of shape is called a bell-shaped curve, and this explains the enzyme action or the effect of temperature on enzyme action. So enzymes work best under optimum temperature, which is roughly around 37 degrees Celsius, which happens to be our body temperature. Okay, so at our body temperature, all the enzymes are working under optimum conditions. Let us look at the next factor, that is effect of pH. Here again there is a bell-shaped curve. Okay, so each enzyme has a bell-shaped curve. That means it has maximum activity at the optimum pH. This is the optimum pH. It has the maximum activity here, and at the extremes of ends on both sides of the optimum pH, the activity is decreased. Okay, now this optimum pH is for most enzymes is around 6 to 8. Okay, similarly our body pH also is normally around 7.35 to 7.45. So the optimum pH of the enzymes, most enzymes, and our body normal pH matches. Okay, so that again enzymes, they work at an optimum condition. Now hydrogen ions, temperature, substrate concentration, they will all influence the optimum pH. Okay. And also different enzymes have different optimum pH. Okay, for example, pepsin is an enzyme which is present in gastric juice in the stomach. So this has an optimum pH of around 2. That means it works best at pH around 2. Similarly, enzyme amylase is a pancreatic enzyme. Okay, it works in alkaline conditions. The optimum pH is around 8, and arginase has an optimum pH around 11. Okay. Next factor is presence of activators. Okay, now when will enzymes get activated? Okay, they get activated in the presence of metals. Okay, so certain metals like potassium, calcium, magnesium, zinc, copper, etc., they can activate enzymes. Okay, now how do they activate enzymes? These metals, they can either activate by binding with the enzyme. Okay, so metal activated enzyme. Now this, when the enzyme binds with the metal, there is a loose bond formation. Okay, so the enzyme binds loosely with the metal, and this metal can also be exchanged with other metals. For example, the enzyme is ATPase, it can bind with calcium also, it can bind with magnesium also. Okay, so and the bond between ATPase and calcium is weak. Okay, so this can be replaced by magnesium. On the other hand, we have metalloenzymes, where the enzyme and the metal they bind strongly. Okay, so the bond between the enzyme and the metal is strong, so that it cannot be broken, and the metal cannot be exchanged. Okay, for example, in the enzyme catalase and oxidase, the metal is iron, so that iron is strongly binding, it is not replaceable. And in case of hexokinase phosphofructokinase enzyme, the metal is magnesium. Okay, so this is the difference between metalloenzymes and metal activated enzymes. Okay, metalloenzymes have strong bonds between the metal and the enzyme, whereas metal activated enzymes have loose bond between the enzyme and the metal. Now, the other mechanism by which enzymes become activated is conversion of the precursor form or the inactive form of enzyme, which is called zymogen or a proenzyme, to the active form of the enzyme. For example, trypsinogen is the inactive form, or the zymogen, it is converted to trypsin by removing a small peptide. Okay, so trypsin is the active form of the enzyme. Okay, similarly, coagulation factors become active form in the of the coagulation factors, from inactive coagulation factors to active forms. Okay, so that is how enzymes get activated, presence of activators. Now, presence of inhibitors. Now, enzyme inhibition itself is a very important topic. Okay, and there are various types of enzyme inhibitors. For example, competitive inhibition, non-competitive type of inhibition, uncompetitive type of inhibition, suicide inhibition, allosteric inhibition, and feedback inhibition. So let us look at one by one. First is competitive inhibition. Now here the inhibitor competes with the substrate to bind with the active site of the enzyme. Okay, so this is the enzyme. Now this is the substrate. Now if you look at the inhibitor and the substrate are structurally same. It is same. Okay, so this inhibitor will compete with the substrate to bind with the active site. Okay, so when the enzyme binds with the substrate, you get enzyme substrate complex. But if the inhibitor comes and binds to the active site, you get this enzyme inhibitor complex, and this does not proceed further to form products. Okay, so the enzyme becomes inhibited. Okay, here enzyme substrate complex, because the substrate is binding to the enzyme, it leads to formation of product. Okay, so here the inhibitor is the structural analog of the substrate. Okay, that means the structure of substrate and the inhibitor is same, as we can see. The velocity of the reaction is same with the substrate. That means Vmax is constant. So the maximum velocity when the enzyme substrate complex is formed, the maximum velocity is same. The affinity of the enzyme to the substrate is decreased. Okay, so the Km value is increased. Okay. Now the enzyme affinity of the enzyme to the substrate is decreased. Why? Because the inhibitor also has a chance of binding with the enzyme. Okay, so the affinity of the enzyme with the substrate is decreased. Okay, so when the affinity is decreased, that means the Km value is increased. Okay. Next we have is the inhibition is reversible if the substrate concentration is increased in the presence of inhibitor. Okay. Now this inhibition, competitive type of inhibition, is reversible. Now when it becomes reversible, when the substrate concentration is increased. Okay. Now let us assume we have lots of substrate concentration. So the chance of substrate binding to the enzyme is more as compared to inhibitor binding to the enzyme active site. Okay, and this leads to leads to the action, the enzyme inhibition becoming reversed. Okay, it becomes reversible. Okay, if you look at the graph, okay, here we have seen that in the presence without the inhibitor we have a Vmax, or the maximum velocity, and in the presence of inhibitor the maximum velocity is same. Okay, so Vmax or the maximum velocity is same without the inhibitor or with the inhibitor, but the Km value in case of inhibitor is increased. Now increased Km value indicates a decreased affinity. Okay, Same with the Lineweaver-Burke plot. You can see over here the Km value is increased, okay, and Vmax remains the same. Now let us look at the various examples for enzyme inhibition, okay, and what clinical significance it has. Okay, so we have the enzyme xanthine oxidase. Okay, now this enzyme is important in uric acid metabolism. Okay, now xanthine oxidase acts on the substrate which is xanthine or hypoxanthine. Now we have the inhibitor allopurinol, which is a substrate analog of hypoxanthine or xanthine, and it inhibits the enzyme xanthine oxidase. Okay, so when xanthine oxidase is inhibited, uric acid formation is decreased. Okay, so this is a drug which is given to decrease the formation of uric acid. So in patients of gout, okay, so gout patients who have gouts, they have high uric acid levels which gets deposited in their joints and results in painful and swollen joint. Okay, most commonly the joint of the great toe. Similarly, we have another enzyme which is steroid synthetase, which acts on the substrate PABA to form is steroid, which is again leads to formation of folic acid. Okay, and now folic acid is necessary for bacteria to work or bacteria to live. Now sulfonamide is an antibiotic. Okay, now this sulfonamide inhibits the enzyme steroid synthetase. Okay, how? Because it's a structural analog of PABA. Okay, so sulfonamide is an inhibitor. It's a structural analog of PABA. Okay, so it inhibits the enzyme steroid synthetase. So when this enzyme is synthetase, steroic acid is not formed, and as a result folic acid is not formed in bacteria. Okay, so that leads to death of the bacteria, or it's a antibiotic which is antibacterial. Okay, so the bacteria needs folic acid for growth, which is synthesized from PABA. Okay, competitive inhibition of PABA inhibits its growth. Next enzyme is dihydrofolate reductase. Dihydrofolate reductase acts on dihydrofolic acid. Okay, now we have inhibitors like methotrexate, trimethoprim, pyrimethamine. So these inhibitors, they compete with the substrate which is dihydrofolic acid and inhibits the enzyme dihydrofolate reductase. Okay, now these drugs, methotrexate is used as an anti-cancer drug because again folic acid is necessary for DNA synthesis. Okay, so if we inhibit the formation of folic acid, then it acts as an anti-cancer drug. Also trimethoprim and pyrimethamine, it acts as an antibacterial. Okay, so in the bacteria, if folic acid is inhibited, it leads to arrest of the growth of bacteria, so it becomes antibacterial. Okay, same as sulfonamides. Okay, next we have other enzymes like vitamin K epoxide reductase, okay, which acts on vitamin K, and we have is dicumarol or warfarin, which inhibits this enzyme. Okay, so vitamin K is necessary for activation of clotting factors, so vitamin K will not act. It leads to inhibition of clotting factors, or it leads to anticoagulant effect. Another enzyme is HMG coenzyme reductase, which acts on HMG coenzyme A and leads to formation of synthesis of cholesterol. Now we have drugs like statins. The group of the drugs is known as statins. The example is atorvastatin, which competitively inhibits HMG coenzyme reductase. So if HMG coenzyme reductase is not working, cholesterol synthesis is lowered. Okay, so this is used to lower the cholesterol levels in the blood. Another important enzyme is pyridoxal kinase. Now pyridoxal kinase acts on the substrate is pyridoxine, okay, and leads to formation of the active form of pyridoxine, that is pyridoxal phosphate. Okay. Now isoniazid is a drug it's an antitubercular drug, which is when given to a patient for treatment of tuberculosis. This isoniazid will compete with pyridoxine and will inhibit the enzyme pyridoxal kinase. So if pyridoxal kinase is inhibited, pyridoxine will not be converted to pyridoxal phosphate, which is the active form of pyridoxine. Okay, so it leads to pyridoxal phosphate deficiency. Okay, so these were the few important clinical importance of the competitive enzyme inhibition. So what you need to remember is these are the examples of competitive inhibition. Okay, so we have inhibitor which is inhibiting a specific enzyme. Now if the substrate concentration is increased, then the effect of inhibitor is nullified. Okay, so it becomes reversible. So please remember this. Next we have is non-competitive inhibition. So in this type of inhibition, as the name suggests, there is no competition between the substrate and the inhibitor. Okay, so we have the enzyme over here, we have the substrate over here. It forms enzyme substrate complex and product. Then if we add an inhibitor over here, okay, so the enzyme substrate, the inhibitor binds to a site which is different from the active site. Okay, so there is no competition between the inhibitor and the substrate. Okay, the inhibitor binds the binding of the inhibitor does not affect the binding of the substrate. Okay, so here affinity of the enzyme and the substrate is not altered, so Km value will remain the same because the affinity of the enzyme and the substrate is not affected, right? So this inhibitor is not affecting the affinity of the substrate to the enzyme, so the Km value remains same. But because it is an inhibition, it is bringing about inhibition of the reaction, so Vmax is reduced in this case. Okay, so the inhibition is generally irreversible and not be relieved on increasing substrate concentration. Okay, unlike the competitive inhibition, where when you increase the substrate concentration, the inhibition is reversible. So here the inhibition is not reversible. Okay, so if you look at the graph, so we have the substrate concentration here and the maximum velocity over here. In the presence of inhibitor, okay, this is the graph which you get. In the absence of inhibitor, this is the graph. So you can see over here, in the presence of inhibitors, the maximum velocity is reduced. However, the Km constant remains the same. If the Km constant remains the same, that means the affinity of enzyme to the substrate remains the same. Okay. Whereas in competitive inhibition, the inhibitor was inhibiting the binding of substrate and the enzyme. So the affinity of substrate and the enzyme was decreased in case of competitive inhibition. So there the Km value was increased. Here the Km value remains the same. Let us look at few clinical examples. Cyanide, which inhibits the enzyme cytochrome oxidase. Okay, how? Because it irreversibly chelates the ferric ions. Okay, so we saw it's a metallic enzyme, cytochrome oxidase, which contains iron. Okay, so if iron is chelated with the help of cyanide, then the enzyme becomes inhibited. Similarly, fluoride, it removes magnesium and manganese from the enzyme enolase and thus inhibiting glycolysis. So fluoride is when we collect blood samples for sugar estimation, the vacutainer or the sample container which we use is a container which contains fluoride. Why? Because this fluoride will inhibit glycolysis, and if we inhibit glycolysis, then the sugar value is not decreased and we get a correct value of sugar. Okay, because if we do not inhibit the enzyme enolase, then the glycolysis will continue in the RBCs and other cells, and the sugar will be utilized and we'll get a false value of sugar. So we want to inhibit the glycolysis. Okay, so that is why we use this fluoride, which is an example of non-competitive inhibition. Then we have it iodoacetate, which inhibits enzyme having SH group at the active site. Similarly, we have is British anti-leucide, okay, which is an antidote for heavy metal poisoning. Why? Because British anti-leucide BAL has numerous SH group. Okay, so the heavy metal also has heavy metal binds with all these SH groups. So the heavy metal, the deleterious effect or the poisonous effect of or the toxic effect of heavy metal does not take place on the enzymes of the body. Okay, so all the heavy metal comes and attaches to British anti-leucide. Why? Because British anti-leucide has numerous SH groups, and the heavy metals, they target the SH groups. Okay, so they bind with the SH groups of British anti-leucide, which acts as an antidote, and not with the SH group of enzymes of the body. okay, then it will have poisoning effect. Another example is disulfiram, which inhibits the enzyme aldehyde dehydrogenase and prevents further oxidation of alcohol and its toxicity. Okay, so the toxicity of alcohol is because of the metabolic product of alcohol, breakdown of alcohol. So so the enzyme which is responsible for breakdown of alcohol is alcohol dehydrogenase, aldehyde dehydrogenase. So if we inhibit the breakdown of alcohol, okay, for example with the help of disulfiram, then it prevents further oxidation of alcohol and its toxicity. Next type of inhibition we have is uncompetitive inhibition. So in this case, the inhibitor binds to the enzyme substrate complex. So inhibitor does not bind to the free enzyme. So in the previous type of inhibition, that is the competitive and as well as non-competitive inhibition, we saw that the inhibitor was binding with the enzyme. Now in case of uncompetitive inhibition, the inhibitor binds with the enzyme substrate complex. Okay, so the enzyme has already binding with the substrate, and then the inhibitor binds to it. So in this case, both the Vmax and Km value both are decreased. We have next is suicide inhibition. It is also called mechanism based inactivation. Okay, that means the inhibitor, the enzyme converts the inhibitor into a product which is more potent inhibitor of the enzyme itself. Okay, it's a type of, again, irreversible inhibition. The structural analog is converted into a more potent inhibitor with the help of enzyme it inhibits. Okay, for example, allopurinol inhibits the enzyme xanthine oxidase. Okay, so we just saw that allopurinol is a competitive inhibition of the inhibitor of the enzyme xanthine oxidase. Okay, so xanthine oxidase is so what it does is it converts allopurinol into alloxanthine. Now this alloxanthine is a more potent inhibitor of xanthine oxidase as compared to allopurinol. Okay, so allopurinol is competitively inhibiting xanthine oxidase. Okay, and also xanthine oxidase, which it converts allopurinol into alloxanthine, which is a more potent inhibitor of xanthine oxidase. So this is an example of a suicide inhibition. Okay. Next we have is allosteric inhibition. So allosteric enzymes have allosteric sites to which modifiers bind. Okay, so allosteric inhibition is seen with allosteric enzymes. Okay, it is not seen with all enzymes, it is seen with only allosteric enzymes. So what are those allosteric enzymes? Allosteric enzymes are those enzymes which have allosteric sites, okay, or a modifying sites to which these modifiers can bind. So we have positive modifiers as well as negative modifiers. So positive modifiers increase the catalytic activity of the enzyme, they increase the enzymatic action, whereas negative modifiers, they decrease the enzymatic action. Okay, so in case of allosteric inhibitor, okay, so the inhibitor is not a substrate analog. Okay, so the inhibitor and the substrate, the structure is different. So allosteric inhibition shows a sigmoid curve, and these type of inhibitors are used as regulatory enzymes. Okay, so you can see over here, so with the positive modifiers, this is the rate of the reaction. Okay, with a negative modifier, so this is the rate of the reaction. So allosteric regulation usually gives a sigmoid shape of curve. Okay. So it may be partially reversible when excess of substrate is added. Okay, so in this case, when the excess of substrate is added, so partial enzymatic action becomes reversible. With respect to the Km value is increased. Okay, that means the affinity of the substrate and the enzyme is decreased. Also the Vmax is decreased. It's a type of inhibition, so the Vmax is decreased. So let us look at the examples of allosteric enzyme. Okay, so there are two enzymes of you can remember, one is phosphofructokinase, the next is acetyl coenzyme carboxylase. Okay, so phosphofructokinase is an enzyme which is present in glycolytic pathway. Okay, so we have activators, allosteric activators is ATP and citrate, whereas the inhibitor is fructose 2,6 bisphosphate. Similarly, acetyl coenzyme carboxylase is an enzyme which is used for fatty acid synthesis. The activator for this enzyme is fatty acyl coenzyme A, okay, whereas the inhibitor is citrate. Last one we have is the feedback inhibition. So it is also known as end product inhibition. Okay, so the end product or the final product of the enzyme inhibits the enzyme. Okay, so for example, the final product of purine synthesis, okay, which is AMP, it inhibits the first step of the pathway. Okay, so for example, A is getting converted to B with the help of enzyme E1, B is getting converted to C with the enzyme of E2, and C is getting converted to D with the help of enzyme E3. So you can see over here the final product pathway D it inhibits the enzyme E1. Okay, so this is feedback inhibition. Okay, so the end product inhibits the first enzyme of the first step of the pathway, for example. Okay, so these are the types of enzyme inhibition that we have discussed. Okay, next is we have induction and repression. So what do we mean by induction and repression? So induction is basically increased synthesis of enzyme at the genetic level. Okay, so when the genes they start functioning more and the enzyme synthesis is increased, that is called induction. Okay, whereas repression is decreased synthesis of enzymes at genetic levels. Okay, so if the genes they decrease the production of enzymes, it is called repression. Okay, induction is increased synthesis, increased genetic production of enzymes. Repression is decreased genetic production of enzymes. The genes for enzymes are induced or activated. Here the genes for the enzymes are repressed or inhibited. Okay, so why will, in case of induction, why will the genes produce more enzymes? Because those genes are activated. Okay, similarly, why will in case of repression, why will the genes decrease the synthesis of enzyme formation? Because these enzymes are repressed or inhibited. Genes are repressed or inhibited. Okay, for example, glucokinase enzyme is induced by insulin. Okay, so insulin acts at the genetic level. It induces or activates the genes which are responsible for production of glucokinase enzyme. Okay, so insulin acts as a positive, or it induces the synthesis of genes which are responsible for glucokinase. Similarly, ALS synthase enzyme is repressed by heme. Okay, so heme formation in case of hemoglobin. So heme is a component of hemoglobin. So increased heme formation will repress the genes which produce the enzyme ALA synthase. Okay, so this is an example of induction and repression. Okay, so what you need to know is induction and repression act at genetic levels. Okay, induction is increased activity of genes leads to increased production of enzymes. Repression is decreased activity of genes leads to decreased production of enzymes. Okay. Then we have is covalent modification. So what do we mean by covalent modification? The activity of the enzymes can be increased or decreased by covalent modification. Okay, so we can also regulate enzymes by covalent modification. Okay, their activity can be monitored or regulated by covalent modification. So what is this? It's a reversible reaction. Okay, example is protein phosphorylation of the enzyme. Okay, now let us look at this particular cascade. Okay, now the hormone, let's say epinephrine or the glucagon, this hormone comes and binds with the receptor. Okay, it activates an enzyme which is called adenylate cyclase, which is present in the cell membrane. Now this enzyme adenylate cyclase will convert ATP into cyclic AMP. Okay, now this cyclic AMP will activate protein kinases which is present in the cell. Okay, so protein kinase becomes activated form of protein kinase. Now this activated form of protein kinase causes catalytic action by phosphorylation. Okay, now this protein kinase will convert phosphorylase kinase to active form of phosphorylase kinase which contains phosphate group. Okay, now this phosphorylated active form of phosphorylated kinase will, let's say, will convert glycogen phosphorylase inactive form to active form of glycogen phosphorylase, okay, which contains the phosphate group. Okay, so this glycogen phosphorylase will cause breakdown of glycogen to glycogen plus glucose 1 phosphate. Okay, so you can see over here is protein phosphorylation of the enzyme. Okay, so we have is phosphorylation of these enzyme can bring about regulation of the end product. Okay, that means regulation of a pathway. Okay, so if we have is, let's say if we dephosphorylate this, then the reaction, this particular enzyme will be inhibited. This particular enzyme will not become activated. Okay, so this cascade, if you see over here, we can regulate this enzymatic cascade with the help of covalent modification or phosphorylation. By addition of phosphate group, removal of phosphate group. Addition of phosphate group will activate the enzyme. Removal of phosphate group will inhibit this enzyme. Okay, on the other hand, okay, here if we have is if we remove the phosphate group, okay, then the enzyme becomes activated. Okay, so again, presence of phosphate groups or removal of phosphate groups can activate one enzyme or it can inhibit another enzyme. Okay, so this is how we regulate or bring about covalent modification of enzyme. Okay, we will study more about this when we talk about metabolism. Next we have is stabilization and compartmentalization. Okay, so stabilization is if the degradation of enzyme is prevented, then its activity is increased. Okay, so if the enzyme is not degraded, obviously, then it has a longer life, so its activity is increased. For example, phosphofructokinase is stabilized by growth hormone. Okay. Second, another way of regulation is by compartmentalization. Certain enzymes may be located in the cytoplasm or it may be located in the mitochondria. Okay, for example, heme synthesis, urea cycle, some enzymes are cytoplasmic and some enzymes are mitochondrial. Okay, and they can help to regulate the pathway. Okay, so that again we will see when we talk about metabolism. So stabilization and compartmentalization also is a way how we affect the rate of the reaction or enzymatic action is affected. Okay, so these thank you very much. These were the various factors which affect enzyme action.